who has joined us today in our second virtual academic conference of biointegration. Today, we are very fortunate to bring to you the topic of frontiers and interdisciplinarity in nanomedicine. So before that, let me introduce myself. My name is Peyer So, and I'm the executive editor of biointegration, and I'm also a professor at Sun Yat-sen Memorial Hospital in Sun Yat-sen University. And let me introduce biointegration. Biointegration is an international open access journal dedicated to multidisciplinary views driving the progress of modern medicine. And biointegration provides a cross-disciplinary platform devoted to foster collaboration in research, education, and communicating crucial advances in various fields. Biointegration offers insights in different areas of life sciences,
Hello?
such as biology, bioengineering, clinical research,
Hello?
public health, medical physics, in order to encourage cooperation and exchange among scientists and clinical researchers. So,
Hello?
yes, Professor John, we can hear you. So now let me introduce the co-editors in chief. We have two co-editors. They are Professor Zhi Chen from Guangzhou Medical University and Professor Pingtong Huang from Zhejiang University. And I'm the executive editor of Biointegration. So if you have any questions or inquiries about how to publish or how to submit your manuscript, or if you want to join us as an editorial board members, please send us an email stated here in the website. As we all know, we are a very new journal. We have our first volume, which is in March 2020. And now we are on our second number of volume 1. So if you would like to know what are the papers that are published, you can actually see it in our website, but I also put it here. We have 6 different publications so far in the first volume. And let me show you some of the highlights in our second issue. So one of that is from Professor Postema. As if you have joined us last time, you will know he was one of our speaker. And also this is the paper. This is from myself and also Professor Xu, who is also the third speaker of today. And this is about enhancing chemotherapy by RNA interference. And also we also have a clinical case report. And this is one of the reasons why we highlight biointegration, because we do encourage people to let us know what kind of lesson we should learn from multidisciplinary integration. So these are some of the papers that we have published so far. And so the purpose of VAC, as we call it, the Virtual Academic Conference, is actually to highlight the establishment of biointegration, to emphasize the importance of integration in today's modern medicine and science, and also to learn from the experts in interdisciplinarity and integration in various fields. And for those who have joined us in June, we already have our first series of VAC, and today we are very proud to come up with our second conference, which is focusing on frontier and interdisciplinarity of nanomedicine. So without further ado, let me introduce today's speaker. We have 4 very renowned professors today with us. And we have Professor Sang-Yong John as our first speaker. We have Professor Jinjun Shi, we have Professor Xiaoling Xu, and Professor Chen Yuan. So welcome to all the speakers. And so, without further ado, let me introduce the first speaker of today, Professor Sang-Yong John. So, Professor John is my PhD advisor and my mentor for life. So, a little bit about Professor John. He received his bachelor, master's, and PhD degree all from KAIST, and so you can see how he loved KAIST. And he's now back in KAIST as a chair professor, and he's also the director of the Center of Precision Bionanomedicine in the Department of Biological Sciences in KAIST. And his research interest actually lies in the interface of biomedicine, biotechnology, biomaterials, and nanomedicine. So he has a lot of very cool ideas. So, and I think it's better for him to introduce himself to you. So today, Professor John will share with us one of his current research interests entitled Integration of Naturally Occurring Bioactive Compounds into Nanomedicine. So let us welcome Professor John. Professor John, can you unmute? Yes, thank you.
All right, can you hear me now?
Yes.
Okay. Anyway, thank you, Payer, for inviting me, as one of our speakers in this wonderful, session. Also, this is my first time experience, to have, to give a talk and virtually, and there's a virtual conference. So, but very nice than I expected. Anyway, so as you know, Peya already introduced me, also introduced the title of my talk. I want to talk about some naturally occurring bioactive compound derived nanoparticle for some biomedical application. So before going into the details of the my talk, I'm gonna please give me a sec, excuse me. My slide doesn't go on next. Okay. All right. So as you know, Pei already introduced, I was trained as a chemist. I received all my degree in organic chemistry at KAIST. But now I'm actually professors in Department of Biological Sciences. And if you see this slide, I received my PhD in organic chemistry, but I did my postdoc in biomaterials sectors under the guidance of the Bob Langer at MIT. So at that time, I switched my research interest from organic chemistry to some biomaterials. I learned a lot, while I was a postdoc in this lab. And then I was appointed as, associate assistant professors in Department of Life Sciences at GIST, Gwangju Institute of Science and Technology. And then I moved to the KAIST, but the similar Department of Biological Sciences. But as you know, even though I was trained as organic chemist, but most of my students, from this biological science, biology. So which means I have to work with them in some biology related some topic as well as some biomedical application. So because of that, I have been working on or collaborating with the some scientists of clinician at hospital in Korea. Also, I've been also very keen interested in translation of my technology into the clinic. So I founded the 3 startup companies so far. So actually, my career itself is kind of some integration of several multidisciplinary some research field. So now currently, I'm working on these 3 main themes: nanomedicine for imaging and therapy, particularly for cancer and inflammatory disease treatment. Or second is some peptide or protein-based biomedicine, that is nanomedicine. Also, we are working on the cancer stem cell related some field as well. But our interest or my interest, has been changed from this topic to other topic here and there. But one thing that wasn't changed, throughout my career was I've been interested in, some developing platform technology which may address so unmet medical need, ultimately, eventually, translation into the clinic. So my main focus has been on development of some platform, okay, or the platform technologies. Let me show you, just several the platform technology we developed. The first platform we developed years ago is novel class of high affinity peptide, which we called peptide, standing for some, aptamer-like peptide. So it looks like, this kind of some bipolar shaped peptide. It has pre-organized structures. Let me I realized I didn't actually turn on my laser pointer. Sorry about that. Now you see the laser pointer here, right? Okay, so this peptide has this kind of pre-organized structures. There, at the center, there is beta hairpin structures. This is a constant region. It's consisting of only 12 amino acids. So forming this kind of very robust and stable beta hairpin structures. And then we introduced two arms at both ends, N terminal and C terminal, and construct these, two arms as library. For example, each six amino acid, so 12 amino acid can be randomized. So we are successfully, able to construct this peptide library. So by using fuzzy display-based technology, you're able to, identify some protein target specific this kind of peptide molecules. So which shows, several tens of nanomolar affinity after just, first, the screening. So by using this target specific peptide, what it did was we used, this cancer, some protein specific peptide as a cancer targeting ligand, introduced them on the nanoparticle surface so that we are able to, achieve, cancer cell specific drug delivery or imaging, application. Also, we are able to fuse the cancer targeting peptide to protein therapeutics, cancer therapeutics, for example, TNF alpha. So this fusion protein was actually very effective in killing cancers. Also, this kind of peptide and antibody, the hybrid form was successfully, formed. So which showed very long-lasting some blood circulation, as well as cancer targeting and very deep penetration into the tumor. Also, peptide, this target-specific peptide per se have been used as therapeutics. For example, STAT3, peptide was used to, control the cancer. So the VEGF-specific peptide has been used to treat, age-related macular degeneration, so-called AMD disease. So anyway, so we are able to use this target-specific peptide to treat various disease. So second, platform technology we developed is polymer thin film platform. So which enable us to convert cancer cell to cancer stem cell spheroid. So once we actually successfully generated, cancer stem cell spheroid from diverse cancer cell source, cancer cell lines, as well as patient-derived primary cancer cell, then what it did was, analysis of a gene expression using RNA-seq, which give us, a variety of the bioinformatics data. Then, based on these bioinformatic data, we are able to, identify some very unique novel biomarkers that can be used for diagnostic marker or therapeutic markers. So we are actively working on this cancer stem cell field because we have the platform technology that generate, cancer stem cell spheroid from just, original cancer cells or patient-derived primary cancer cell. It's a very unique technology we have. Also, the last one is the bilirubin-based nanoparticles. But I'm gonna, explain this because this one is the main theme of my talk today. So I'm gonna show you. So let me back to the today's main theme. So, which is integration of a naturally occurring bioactive compound into nanomedicine. So, as you know, there are many different types of, naturally occurring bioactive compounds. Shown here is just a few of them. Just name a few. For example, curcumin, active ingredient from the curry, Indian curry. Also, resveratrol, which is active ingredient from wine. EGCG from green tea, and bilirubin, which is endogenous, antioxidant molecules. So we produce the bilirubin every day with a high amount. But anyway, the problem with this, the naturally occurring bioactive compounds is, even though, these bioactive compounds actually have demonstrated therapeutic potential in many disease models, but none of these actually successfully translated into clinic. So why is that? So we wonder, why is that? So as you know, all these bioactive compounds are poorly water soluble. Because of that, after, for example, oral administration, their bioavailability into the bloodstream is very, very low, less than 1%. Okay. Also, all these, if you look at this chemical structure, I'm organic chemist. So the chemical structure, you will easily find they have many conjugated double bond, which means they intrinsically very sensitive to some environment, which means light sensitive, which means unstable. So because of this property, it's very hard for scientists to translate them into the clinic. So how to overcome this intrinsic problem or associated some limitation? One approach may be encapsulating these bioactive compounds into some nanoparticle or liposome, right? Or you can make this some amphiphilic version of bioactive compound so that they are self-assembled into nanoparticle. Okay, so there are the pros and cons these two systems. In this case, that merely encapsulate this bioactive compound in the core of a nanoparticle. Of course, you can increase the systemic stability, water solubility. The problem with this system is, the loading capacity of a bioactive compound per nanoparticle carrier is very low. Let's say less than 10% or something like that. Also, nanoparticle carrier per se, the carrier itself may have some potential toxicity, right? But if you look at the other side, this bioactive compound derived nanoparticle, which consists of only entirely consisted of this bioactive compound. For example, polyethylene glycol modified bioactive compound can self-assemble into these nanostructures, right? In this case, nanoparticle per se can act as therapeutic molecules, as well as the carrier molecules for many different types of cargo, therapeutic cargo. Okay. So I'm going to show you, one typical example, what we actually overcome those some bioactive compound and related some the limitation using this pegylated bilirubin. So we developed pegylated bilirubin derived nanoparticle and then use them for treatment of anti-cancer, cancer or inflammatory disease. So I have the conflict of interest disclosure because I founded a startup company 2018, the Billix, acting as co-founder as a board member. So because Billix is actually founded based on the this pegylated bilirubin technology. Anyway, so let me give you just a brief introduction. What is a bilirubin? So bilirubin is actually, final metabolite of a heme. As you see, the bilirubin is a final metabolite, and also the endogenous antioxidant compound is one of a bile acid. Yeah? So we produce this bilirubin, is a very large amount. So 10 milligram, approximately 10 milligram per liter of blood. So which means the bilirubin may function, biological function, important function, play very important function. But if you have too much, bilirubin in your bloodstream, you will have, jaundice signature because this bilirubin, which is very hydrophobic, is insoluble in water at all. So this actually must be excreted from liver from this, some metabolism pathway, so this enzyme. But if you have too much bilirubin under some disease condition, such as some liver disease, okay, then it cannot be excreted from liver, then become deposited in various tissue, including skin and white scleral part of eyeball. So you'll have this jaundice signature. But this jaundice signature is not a disease. It's kind of some sign you have some problem with your liver or in your body. Anyway, so there have been, some many epidemiological study on the relationship between, serum bilirubin level on and some disease condition. So anyway, to make long story short, so these people who have high bilirubin level experience a much lower risk of having inflammatory disease as well as cancer. Anyway, so what is that? Because bilirubin, it turns out bilirubin is a very potent antioxidant and also anti-inflammatory agent. So which can scavenge, various types of reactive oxygen species in our body, thereby protecting our tissue and cells from this oxidative damage. Okay? Because of that, scientists actually already, confirmed the therapeutic efficacy of bilirubin and in many different types of inflammatory disease and also metabolic disease animal model, in which bilirubin is turned out to be very effective, potent therapeutic molecules. So now you understand, bilirubin has Jekyll and Hyde character, double-edged sword. So we wondered how we were able to, get rid of this Hyde character specifically, then so that we can, utilize that, Jekyll is good guy's property of bilirubin. So this was very simple solution because we just attached polyethylene glycol, one of these two carboxylic acid, then preparing synthesized monopegylated bilirubin, which is amphiphilic. So self-assembled into nanoparticle in aqueous medium, as you see here. So as you know, reactive oxygen species, play a crucial role in progression and pathogenesis of many different types of the some inflammatory disease, either chronic or acute inflammatory disease. So we actually tested the therapeutic efficacy of our bilirubin nanoparticle in, for example, here, some inflammatory bowel disease model. As you see here, bilirubin nanoparticle treatment actually prevent the progression and pathogenesis of IBD very effectively. Also, the in hepatic ischemia reperfusion injury model, which can be prepared by microclamping at this, vessel, so that 2/3 of the liver fell into the ischemia state. Anyway, in this model, our bilirubin nanoparticle administration through intravenous injection showed actually normalized the ALT and AST level close to normal compared to untreated control group, which showed a very high increase in the ALT, AST. Also, bilirubin nanoparticle were able to prevent some hepatic damage, HNE staining and tunnel assay, so apoptosis is very effectively prevented. Also, necrotic area was way high reduced upon this bilirubin nanoparticle treatment. Anyway, so in cardiac ischemia perfusion injury, which is very close to the hepatic injury model in terms of the mode of action, so which was done by Professor Peter Gang at Harvard Medical School, so clinician and cardiology clinician, according to his experiment data. So bilirubin nanoparticle treatment actually back to the normal function of the cardiac. So it is very effective, potent in preventing this cardiac IRI as well. So anyway, so due to the time limitation, I'm gonna just show you this bilirubin nanoparticle. We showed bilirubin nanoparticle could be used as universal anti-inflammatory therapeutic nanomedicine without immunosuppression. So we already showed the therapeutic efficacy in this islet transplantation model, hepatic IRI, acute asthma, also the after oral administration of hyaluronic acid bilirubin conjugate, which successfully inhibit the progression of IBD after oral administration. Also, the not only this disease, but also the psoriasis, multiple sclerosis, autoimmune disease model, we show the very potent therapeutic efficacy of our bilirubin nanoparticle nanomedicine. So you may wonder why this bilirubin nanoparticle showed a very potent therapeutic efficacy compared to just free bilirubin or other antioxidant agent. Here there are several reasons, but I'm gonna show you just the three key actually aspects of bilirubin nanoparticle have. First, this is an N-acetylcysteine is a NAC. It's FDA approved antioxidant drug, approved to treat acetaminophen induced acute liver failure. It's FDA approved antioxidant. Compared to this NAC, bilirubin nanoparticle is 100 times fold highly more antioxidant power it has. Okay. Also, because the bilirubin nanoparticle is a nanoparticle, pegylated version, it can long circulate in the bloodstream when it injected. So as you see here, the half-life of bilirubin nanoparticle is almost 7 to 9 hours, which is way longer than small molecule, which mostly shows just less than 30 minutes or 15 minutes in blood circulation. This is second reason. Third reason, because of this nanoparticle character, it has ability to passively target the inflammatory site where some vascular permeability is increased, right? So bilirubin nanoparticle preferentially accumulate, of course, in a passive manner, not active manner, inflammation site. So for all these three reasons, we think the bilirubin nanoparticle showed very potent therapeutic efficacy. It's almost, so what about safety? So we confirm, bilirubin nanoparticle has any lethal toxicity, and apparent, body weight measurement or hematological or histological level, there we didn't see any, toxicity after we injected, 60 times higher concentration than the level in our body endogenous level of bilirubin. It's very safe. The other aspect, of a bilirubin nanoparticle has, it can be used as, some therapeutic medicine to treat inflammatory disease, but also can be used as a direct delivery carrier. But because of time limitation, I'm going to show you this bilirubin nanoparticle. Once, irradiated, it converted to some water-soluble version of isomer is suddenly disrupted and releasing drug, the environment. Also, interestingly, this bilirubin particle is once oxidized when they encounter, reactive oxygen species, it converted to water-soluble version of biliverdin, which means this bilirubin particle once encounter ROS in our body, endogenous ROS in some disease site, it converted to water-soluble biliverdin, it also get disrupted and releasing drug outside in this manner. So to test that, we actually constructed this biotin-related bilirubin nanoparticle encapsulating doxorubicin, and so we can arrive the tumor microenvironment where the ROS is enriched. Then they react with the ROS, and the particle is disrupted, and they're releasing drug, and then released drug can diffuse into the tumor site more deeply. So because of that, we actually observed very high level of therapeutic efficacy, as you see here. Anyway, bilirubin nanoparticle also, can be constructed as a theranostic, probe, because this pegylated bilirubin, because of this, is endogenous some characteristic to able to, chelate many different types of metal ion. So pegylated bilirubin can successfully coat some metal nanoparticle, surface of a metal nanoparticle, or some metal ion can be easily chelated into this bilirubin nanoparticle layer. Also, some perfluoropentane ultrasonic, imaging gas can be also encapsulated in this manner. So we are successfully able to construct some therapy because of this bilirubin anti-inflammatory agent. Also, the imaging because of this kind of, ultrasonic contrast agent gas or copper 64 or iron oxide nanoparticle for MRI contrast agent. So we are successfully, able to construct this kind of some theranostic, probe. Anyway, so let me summarize my talk. Bilirubin-based nanomedicine. This is because of this very potent antioxidant, anti-inflammatory activity of bilirubin itself. So this bilirubin-based nanomedicine was very effective in treatment of various inflammatory disease. Also, this because of the metaculation ability, so theranostic application was actually possible with this bilirubin nanoparticle. So novel therapeutics for hard to treat inflammatory disease was developed based on this bilirubin-based nanomedicine. And also we are able to some encapsulate the drug molecule additionally to this bilirubin nanoparticle so that they were acting as some stimuli, ROS or light responsive direct delivery carrier, as well as, nano carrier itself acting as, therapeutic molecules. So this is my group. The building, the backside is a newly built some library archived. Actually, Payer actually took a picture with me when she actually visited archived. I don't know when, maybe 2 years ago, 1 year ago. Anyway, so at that time, we took a picture together here, the same, area. Anyway, thank you very much again for joining with us despite your, the very busy schedule. Thank you. Any questions? I will happy to ask, take any question from the audience.
Okay, so thank you, Professor John. Actually, today we will not have any questions from the audience, but
Oh, I see.
yes, yes. But yeah, it's good to see you again. And yes, Professor John is my mentor, and he will be my mentor for life. And this is it's very good to see all the students and all the colleagues before, and I do see some old friends and new faces there. So yeah, we are very, very interested in to see that how a simple idea of making something that is intrinsic to us, something that we have in the body, like bilirubin, and then you have an idea and then you make it into a nanoparticle. So this is, I think, something that is for people who wants to come into nano. Nano is not some very far away or very far-fetched idea. It is actually very simple and it's very doable. So if
Yes,
you
right. Yeah.
yes, so if you have any idea, just come and talk to us. We can help you out there. So thank you, Professor John. And
Right.
we are going into the next speaker, which is Professor Jinjun Shi. So
All
let me
right.
introduce Professor Jinjun. So Professor Shi is an associate professor at Harvard Medical School and is a faculty member in the Center of Nanomedicine and Department of Anesthesiology at Brigham and Women's Hospital. Actually, Professor Xiaoding and I were both actually working together with Professor Shi before we came to Guangzhou about 2 years ago. And so his research involves a highly interdisciplinary combination of nanotechnology, biomaterials, drug delivery, and immunotherapy for biomedical applications in cancer, atherosclerosis, and other diseases. And his current research includes RNA nanotechnology for gene silencing, mRNA delivery for protein restoration, and nanoparticle-mediated immunotherapy. So today, Professor Shi will grace us with his lecture entitled RNA Nanomedicine, Integration of Nanotechnology, RNA Delivery, and Biology. So let's welcome him.
Oh, well, can you hear me here?
Yes.
Okay, well, first of all, thank you so much for having me here today. And it's a great pleasure to see you, to all the friends, to see Professor John. We have been friends for many years, and Professor Xiaoding Xu, and also a new friend, Dr. Yu from Macau. So it's really a great honor to be here today. And then also, I hope everybody is well and safe because of the COVID pandemic. And that's why we have this virtual meeting here today. And actually, but be honest, it's the same to me. And it's also my first virtual meeting to I assume a lot of audience here. So today I will go through first. Please allow me to have a couple minutes to introduce the Center for Nanomedicine at Brigham and Women's Hospital in Harvard Medical School. We are in the Boston area, and this is if you came to Boston before, or you're familiar with Boston, and then you like soccer, like baseball, and then we are in the Fenway area, and then we have the this area for Red Sox team. And this is our kind of new building. It's been like almost 4 or 5 years old. And then we are on the 7th floor of this called the Hill building for transformative medicine. So that the aim of this building is to really develop transformative materials and medicines for patients. And this is a mixture building either with the clinic and research. So the center was actually established by also my mentor, Dr. Omid Faloxad, and also actually the mentor for Pei and then Xiaoding. So now we have 3 labs in Dr. Faloxad's lab, which focus on, as you know, he has done a lot of work with the nanomedicine and a lot of translation work he has done, including biotherapeutics, and also he's very interesting recently on nanobio interactions, and has another company called Seer, and to work utilize the nanobio interactions to study the proteomics. And also we have another lab here. It's called Dr. Jeffrey Cobb's lab, and he's actually not in our department, but in the Department of Medicine. And his lab is focused a lot on the biomimic materials like adhesives, devices. Also, he works with stem cell engineering, and he also has started probably 6-7 companies. And one of them went to public a year ago, and then the other one you probably heard about that is called a Gecko, and to put the adhesive gel on the as a adhesive materials to heal the injury, or the surgery after surgery of the heart. And it's actually in phase 2 clinical trials. And also, Sen has I started my lab about 7 years ago, and our research is focused on mobile responsive nanotechnology, and for particularly biomicromolecule delivery like siRNA or messenger RNA. And we're also certainly interested in immunotherapy, which is hard these days because of the success of PD-1, PD-L1, and then CAR T cells. Also, we're gonna expand this center, and we'll probably soon have another new lab joining the center. So today we'll briefly go through some of our research, but before that, let me first of all introduce overview of what we are doing. And as I mentioned, we are very interested in developing, well, not entirely new, but more rational materials in nanotechnology, and which hopefully we can solve a lot of challenging issues in the systemic delivery of biomicromolecules such as small interfering RNA, messenger RNA, and protein. That's why our main interest in the lab here. We like big molecules instead of small molecules. And so hopefully my talk is somehow very different from Professor John's talk, which is fantastic. And our biomedical application is also broad, but the main focus is on the cancer and atherosclerosis. And more recently, we are also interested in something else like pain medicine too. And meanwhile, we're thinking fundamental research is critical for our design of biomaterials and nanoparticle. So we certainly would like to understand more about how nanoparticle interact with different biological systems, such as serum protein, because this is the first step, right? When you inject a nanoparticle through IV intravenous injection, the first thing nanoparticle nanomedicine met or sees is the serum protein. So this interaction is critical, and how this interaction interact with the pharmacokinetics, biodistribution, efficacy, penetration, etc., all critical, right? So we think the fundamental understanding of the nanobio interaction will help us to develop a more rational or more effective technology or materials. But because for the sake of time today, I won't have much time to talk about the fundamental studies. We'll mainly focus on today's talk on the recent work on developing RNA technologies, including both siRNA and messenger RNA for treatment of cancer and atherosclerosis. I'll give you 3 examples. So let me start first with cancer. So cancer, as we know, is a very complex disease. Everybody knows that it's hard to treat. It's always come back, if you initially have a successive response. And majority of cancers we know involved with the genetic mutations. Compared, if you look at these ACR figure here, compared with normal cells, as you can see, the normal cells, everything's balanced in between the oncogene and tumor suppressor. But when this balance is broken, you have the overexpression of oncogene or more broad the pro-tumor genetic genes, and then the loss of function of tumor suppressor either through mutation or complete deletion. And then this balance is broken. Then the normal cells become tumorigenic. It becomes the tumor cells, and then later on it will metastasize to other places. Also, these genetic mutations can cause the drug resistance. So that's why in many cases the initial treatment was successful, but later on the cancer relapsed. So in current, we see majority of pharmaceutical companies, they are focusing on the targeting of pro-tumor genetic drivers, and many drugs have been successful, including more recently the precision drugs. But unfortunately, still there are a lot of pro-tumor genetic oncogenes. They are considered as undruggable or difficult to drug. Either, it's hard to design small molecules to target these therapeutic potentially proteins, such as the transcription factors. And so that's why one thing we think, RNA interference technology could be very helpful to potentially to target those difficult or undruggable proteins. And on the other hand, tumor suppressors, in many cases for the cancer, they are either depleted completely or mutated. So there are very limited strategies we can do to tumor suppressor. There are certainly some small molecule development to target the upstream or downstream pathways. But if you want to completely restore the tumor suppressors, certainly there are very limited options. And unfortunately, so far, I don't see any FDA approved or clinically approved therapeutics to target tumor suppressors. So that's why we think, using messenger RNA could potentially provide an option for this lost tumor suppressor function. So let me start with the first technology I would like to introduce to you. It's called RNA interference technology, RNAi. And I'm sure many of you know this technology. It's been a while. First discovered in 1998 by two scientists, Andrew Fire and Mellon. And one of them actually is in the Boston area, Craig Mellon in the UMass Medical School. It's about 1 hour driving from Boston. And as you know, on the left side, we can see this is a simple schematic of how RNA technology works. Either, the long strand, double strand RNA in the tumor cells can be degraded by the dicer, the enzyme, into the small interferon RNA. And the small interferon RNA integrated to this ARGO2 RISC, because you got RNAi silencing complex. These are protein complex together, and then the sense strand will be cleaved, and the antisense strand together with this RNAi assembly will target specifically the mRNA. So depending on the sequence, you can almost target any gene you want. So it's a very potent technology, and when it was discovered, particularly after, in 2001, after the report of synthetic siRNA, so many pharmaceutical companies have had a lot of enthusiasm for this technology, because you can specifically target the gene and then inhibit the protein expression. So you can target almost anything you want. And then right after that, 5 years later, they both, Andrew and Craig, won the Nobel Prize in Physiology and Medicine. And then it took 12 years, another 12 years. So in total, you can see from 1998 to 2018, 20 years exactly. We saw the first FDA-approved siRNA therapy for a rare genetic disease called HATTR amyloidosis, which was developed actually in Cambridge, in Massachusetts, by a company called Alnylam. So it took like 20 years from the discovery to get the first FDA-approved drug. It's reasonably long time, but it's also a reasonably good amount of time. The reason is because small interferon RNA has some intrinsic features, such as large biomolecules. Its size is certainly much larger than small molecules. And also it's because it's negatively charged and highly hydrated. So it's really challenging for this siRNA to cross the cell membrane, which is also negatively charged in many cases, to cross the cell membrane and enter into the cytosol. And also the siRNA, as we know, could be easily degraded by nucleus. But I would like to point out here, this is probably, the third point is probably not the big issue anymore these days, because of the advance of a lot of useful chemical modification strategy to stabilize the siRNA or other nucleus. And the fourth one, the challenge is actually, or the features for siRNA delivery is we have to make sure we transport the siRNA into the cytosol, where this RNAi silencing strategy, pathway worked. So I think, here we go. So over the last 10 years, since 2010, when I was a postdoc in Omi's lab, and then we have been keeping developing different nanoparticle technology to trying to solve the systemic isoionic delivery to the solid tumor in particular. So we've been working for many years and then develop a variety of different strategies, including some strategy developed by Professor Xiaoding Xu. And the aim of this, our platform technology, are actually to solve many challenges in the systemic isoionic delivery, including long broad secretion. How can we improve the broad secretion? As we know, a small isoionic by itself it's very, can very quickly degraded by, in the blood, it takes probably like 30 minutes. It's like small molecules. And also the second challenge is how can we improve the tumor accumulation? And the third one is actually is far more important, and then we gradually realize it's critical for the cancer nanomedicine field is how this nanoparticle can penetrate through the tumor tissues and to reach out to as many tumor cells as possible. And then also we certainly want more, cell uptake, and no particular specific cell uptake by either tumor cells or other tumor cells in the tumor tissues. And then endosomal escape is another critical thing, and then that's how nanoparticle can help. And certainly control release, how can we get a sustained silencing is also another challenge. So we have developed this technology to solve, I would say, some of these issues at one time, but not yet, as to my understanding, is to solve all these problems at once. But we're, getting very close to this, and then hopefully next time I can report to you. I can share with you new technology we're developing now to solve all these problems at the same time. So I will just give you one example to how this technology can be used for cancer treatment. So we've been collaborating with Dr. Bruce Zeta from Children's Hospital in Boston. And his laboratory discovered an interesting protein called proheptin-1. And in the clinical sample analysis, they found in later stage of non-small cell lung cancer, high expression of PHB1 is highly correlated with the low overall survival of non-small cell lung cancer patient. So, but unfortunately, there are no specific small molecule inhibitors that people can use to target proheptin-1. So when I talk with Dr. Zeta, we think the RNA technology is perfectly fit to evaluate these targets, say whether this PHB1 target could be potentially used for cancer treatment. So we work together for many years already. And initially, we designed the nanoparticle called the lipid polymer hybrid nanoparticle system. Later on, I'll explain a little bit more about the platform for mRNA delivery. And with the control nanoparticles, as you can see here, so you don't get any silencing. By the way, the red staining is the PHP1. And with the nanoparticle loaded with siRNA PHP1, you can see almost complete silencing of the target. And with the silencing, of course, you can see the apoptosis and very clear apoptosis after the nanoparticle, siPHB1 nanoparticle treatment. And when we do the in vivo intravenous injection, we can see the nanoparticle itself is pretty potent by inhibiting the tumor growth. Also, we can combine this nanoparticle with small interferon PHB1 together with platinum drug, which is the most widely used chemo drug for cancer treatment, including for the non-small cell lung cancer. So we can even get an even better anti-tumor effect. So that's one example, and we have been working on this for many, and collaborating with many other scientists and physicians in this field. So hopefully we'll get something more meaningful done soon. And then I will move to the second topic, the second technology, which is messenger RNA technology for protein restoration, protein replacement. Well, you know probably a lot about messenger RNA now and heard about all the time because of COVID. And then again, another company in the Cambridge side called Moderna developed the messenger RNA COVID vaccine, and which is I think it's about late last week they moved that to the phase 3 clinical trial. So hopefully we will have successful COVID mRNA vaccine soon, and to help the patient, and to hopefully can solve the pandemic issue. So compared with DNA, both messenger RNA and DNA can be used for protein expression, right? So but mRNA is certainly in the middle, and you do the DNA transcription to mRNA and mRNA translation to protein. So compared with gene therapy, which is based on DNA, we think using messenger RNA has several benefits. On the right side, as you can see, first of all, there's no nuclear barrier, so you just need to deliver mRNA to cytosol, not necessarily to the nuclear, like what we have to do for DNA. And also, you can, because of that, then you can avoid the host genome integration, so you avoid any potential toxicity issues. And the mRNA mediated protein expression is faster and more predictable compared with DNA. Also, because of the robust chemical modification, now we can see have a very well, we can have the synthetic messenger RNA with much reduced the immune response and better stability and transfection efficiency. But overall, similar to siRNA, and the key challenge is still how can we deliver the messenger RNA into the cytosol. So the cytosolic delivery is certainly a big challenge. And that's why a lot of different strategies, particularly in lipid nanoparticle technology, have been developed to solve this potential issue. And so in our lab, we started working with messenger RNA about 6-7 years ago. Indeed, we first, when Moderna started the company a couple of years later on, we realized this field could be, very interesting, and then could solve a lot of potential problems. So that's why we started working on this field. And then we certainly did not develop, entirely new nanoparticles for the mRNA delivery. Instead, we have done a lot with the lipid polymer hybrid nanoparticle system for siRNA, and then we transform the technology for messenger RNA delivery. So this is a very simple formulation steps. First of all, we've complexed this messenger RNA with a cationic lipid-like materials. We call it here the GCOC14, which is the generation 0 of PAMAN, polyamidoamine, and we modified it with the hydrocarbon tails, 14 carbon tails. And then this is lipophilic lipid-like materials, then complex with mRNA through the charging interaction. Also in the organic solution, we did dissolve the polymer called the polylactic galactic acid, PLGA. And then we added this DMF solution into water. And then because of the DMF solution is miscible with the water, so the polymer PLGA, which is hydrophobic, and then we'll precipitate it out and together with this complex. And as you can imagine, this mRNA GCOC14 complex, they sort of like have this structure, which we don't know yet whether it's really is a reverse micelle structure or not, but then through these lipophilic or more hydrophobic tails interaction with PLGA, this complex can be trapped in this PLGA core. And on the surface, we also dissolve this lipid pack on the in the solution, in water solution, and then this lipid pack can then self-assemble on the nanoparticles to stabilize the nanoparticle, and also that also gave the nanoparticles long circulation in the blood. So we did a lot of characterization of the nanoparticles, and you can see the size about this mRNA nanoparticle. It's about 100 to 120 nanometer, and it's quite stable in different mediums, including in serum. And we tested the toxicity of the nanoparticle, particularly, most of the concentrations we tested, we don't see obvious toxicity. And then also we compared with a commercially available transfection agent called lipofectamine 2000. Here we believe it is L2K mRNA. And then mRNA nanoparticles show comparable, even better in high concentration with the in terms of transfection efficiency in the tumor cells. So with this platform, we think what we're going to do next, right? So and then when we started this project, first we wanted to actually do vaccines, but then we really realized there's a very interesting niche people have not realized a lot. Is the actually I talked about it earlier, the tumor suppressor loss of function issues in the tumor in cancers. So first the target we pick it up is called P10. Well, why in the prostate cancer? So why we're interested in this is because in later stage prostate cancer patients, there's about 40 to 50% patients, they have this loss of the P10 function. So we think this is going to be great for as a proof of concept study. So we use these P10 mRNA nanoparticles to transfect the P10 non-prostate cancer cells like PC3 cells. So you can see here this green indicated, which is actually hemagglutinin, but it's tagged to P10. So you see the green means you see the P10 expression. Very, very nice expression by our nanoparticles, and then that can induce actually very good apoptosis of the PC3 cells. And we did a Western blot, and you can see the main biological pathway of the P10 resolution is the inhibition of a PI3K-AKT pathway. And in this case, we can see the dose-dependent toxicity of P10 mRNA nanoparticles. Also, we did another P10 loss to prostate cancer cell line called LINCAP. Similarly, we can see this effect. And very interestingly, I didn't put the data here, is if you look at the P10 expressing or P10 normal wild type prostate cancer cells or normal cells, you don't see much toxicity at all after the P10 transfection. And then we move on to study whether this nanoparticle can be used to treat the tumor in vivo. And we established a model called the intratibial tumor model. The reason is because in about 80 to 90% of later stage prostate cancer, they have bone metastasis. So that's why we're interested in the bone tumor, in the tumor in the bone tissue, see whether we can deliver the mRNA nanoparticles to there. So we did the injected the PC3 cells, which is luciferase expressing PC3 cells, into the tibia, and then we monitor the tumor growth using IVS imaging system. As you can see, after treatment of nanoparticles, we do see the inhibition of the luminescence signal as compared with the control groups. So we are pretty happy with this, and we are actually also working these P10 mRNA nanoparticle in combination with other therapies. And the next target we are interested in is actually P53, which is probably more popular, even probably is the most popular tumor suppressor in the area, in the field. And then we want to see whether the restoration of tumor suppressor, particularly like P53 here in this case, whether we can improve the efficacy or sensitize other therapeutics like chemotherapeutics. So we pick up this called mTOR inhibitor everolimus. Everolimus is an FDA-approved drug indeed for certain type of cancers, but not for other popular, more widely, distributed cancers like lung cancer, like liver cancer. Certainly, the failure of the clinical trial of everolimus in these type of cancers certainly is very complicated, and lots of biological studies have been done in the field. And one potential biological mechanism underlying the failure of the everolimus or underlying the heterogeneous response of everolimus to different cancers is because of the activation of autophagy. And then the autophagy activation by the N2 inhibitor can induce a protection of the tumor cells. And then particularly in the P53 lost cancer cells, we also see these anti-apoptosis, also see the cell proliferation, cell cycle loss of control. So all these issues are associated with the P53 lost tumor cells. So very interestingly, we all know P53 has a lot of functions, right? And one particular function is they can induce the apoptosis, they can induce the cell cycle rest. This is all well known, but also there's another unique feature of P53 in the cytosol, not the nucleus P53, but the cytosolic P53, that can actually, overexpressing that P53 can inhibit the autophagy pathway activation. So we think if we can combine P53 nanoparticles together with N2 inhibitor, we probably can see a very potent anti-tumor effect. So that's what we did this, with the hypothesis, we did the test of the P53 mRNA nanoparticles together with everolimus in P53 lost the tumor cell, particularly lung cancer cells and liver cancer cells. So we can see here in this Western blot data, if you can see with only everolimus itself, it actually inhibits the mTOR, as we can see, the phosphorylation. Does induce the autophagy activation, looking at BACKLIN1 and MCLT3-2 expression. If we just look at the lane 2, the P53 mRNA nanoparticle itself, we can see it doesn't affect this autophagy pathway, but here, and because it's not activated, but it does induce the anti-tumor, anti-apoptosis. You can see the downregulation of the BCL2 pathway, and then the inducing apoptosis. But if you combine these two together, you can see here very clearly, it does inhibit the activation of the autophagy pathway. You can see here the last obvious band of the BACKLIN1 and the LC32. And then also, meanwhile, it keeps the anti-apoptosis effect. You can see the downregulation of the BCL2. But then at the synthetic effect, we can see more apoptosis from the tumor cells. So on the right side, you can see the combination did induce much higher cell toxicity and then much higher apoptosis. So we did this in cancer, liver cancer and lung cancer models. So this is just one piece of data I'd like to show you here. By the way, this drug is everolimus is auto drug, so auto available. So you can give auto gavage into mouse, and then but by itself it doesn't induce much effect, anti-tumor effect. And for our P53 mRNA nanoparticle, we give an IV injection, and you can see here that does induce the inhibition, similar to what we saw in P10 study. But when you combine these two together, you can see much better anti-tumor effect. And because of the time limit, I think I need to quickly move to the last topic I would like to share with you, atherosclerosis. As you can see, cancer and atherosclerosis have actually a lot of similarities, because for number one, both tissues are leaky. So you can see cancer is leaky because of inflammation. And again, similarly to atherosclerosis, also because of inflammation. And then the endothelial cells on the plaque tissue of the atherosclerosis is also leaky. So nanoparticle can certainly passively cross these leaky gaps and enter into the diseased tissues. And another similarity I would see is also for atherosclerosis, genetic variation highly involved. And one type of cell type is very critical in atherosclerosis is called a macrophage. So the macrophages, the job the macrophages cells they're supposed to do is to actually enter into these plaque tissues. They eat these dead cells, the small necrotic core, and then supposedly they to reduce the core area. So this is a process called aphelocytosis. But instead, in reality, the macrophage cells did actually the opposite way. So they did not eat much of these debris cells or the necrotic cells. Instead, they actually become more inflammatory. So they induce more inflammation and check more macrophage cells on other immune cells into the plaque tissues, and then increase the plaque size. And then, eventually it will reduce the rupture of the plaque, and then induce a thrombosis and heart attack, as is the end of story. So what we think, with using siRNA nanoparticles could potentially to target the macrophage, and then they inhibit the specific pathways in lesional macrophage, particularly the inflammatory pathways. So one pathway, our collaborator Dr. Eric Bates from Columbia University, his lab discovered the pathway called the CAMK2 gamma pathway. It's a calcium modeling kinase 2 gamma pathway. And in human tissues and mouse tissues, his lab found, in both cases for atherosclerosis, the CAMK2 gamma protein are highly more overexpressed. So we think if we can target the CAMK2 gamma using siRNA nanoparticles, or whether we can induce any effect, therapeutic effect for atherosclerosis. So that's why we designed a very similar nanoparticles, and using, again, this is called lipid polymer hybrid nanoparticles. You can see we don't change much of the components. Again, you use the same cationic lipid-like materials. We're also using a DSP pack here. The difference here, we added a specific ligand called the stabilin 2 peptide to the DSP pack, which you can specifically target lesional macrophage. And also we have this POGA core in the middle, and then siRNA trapped inside of the nanoparticles. So we did a in vitro, in vivo testing to see whether these S2P peptide targeting nanoparticles can have more macrophage recognition. So that's what we did here in vitro studies. You can see, yes, when we increase the density of the S2P ligand on the nanoparticle surface, we did get better cell uptake or more positive staining of the macrophage. Also, we did in vivo studies compare with non-targeted nanoparticles to targeted nanoparticles. We can see about twofold increasing of the accumulation of nanoparticles in the atherosclerotic tissue, and particularly to the macrophage cells. So then we want to see whether this CAN-K2 gamma could be silenced, and then whether the silencing can induce the reduction of necrotic core area. So that's what the result we got, and pretty positive. You can see here after few injections of the SI CAN-K2 gamma nanoparticles by intravenous injection, we can get about like 60% silencing of this targeted protein. That can also induce the overexpression of a downstream protein called the MERTK, which is actually a good protein for atherosclerosis. As you can see here is a tissue staining of the CAM-K2 gamma in the macrophage cells. You can see here, similarly, we can get a 60 to 70% silencing of the protein expression. And this actually led to the reduction of the necrotic core area. As you can see here, this is an area size, and this is two control groups. So, and then certainly we have a lot of other data. So if you're interested, you can take a look at we just recently published a paper in Science Translational Medicine. With this, I would think I will probably end my talk. I'm sorry, I probably took a little bit more time than I supposed to, but I would like to emphasize here is we all know research for any area, there's a research gaps, and then it's very important, particularly for, for similarly, I would say to RNA medicine field, that we first of all need to figure out what disease and cell type we want to target, and then what biological proteins you want to target. And then also we need to understand what the delivery challenging is for the RNA delivery to the disease or to a cell type. And certainly nanotechnology or other technologies, I would say more broadly, and how can we use these technologies to solve the delivery issues, to solve the the challenging the delivery barrier. So, and then we can design a successful product, and move these forward to clinical studies, right? And also we certainly need to consider the formulation, scale up, manufacturing, toxicity of all these technologies. But long and short is we all think, integration is highly important. And I think, this journal biointegration, Pei'er, and then her colleague started, is a very good platform for these, multidisciplinary studies. So certainly I've already, I assume, I would expect that this journal will be highly impactful soon. With this, I'd like to thank, my collaborators, particularly Dr. Eric Tabase for the atherosclerosis work, Bruce Zeta for the siRNA and P10 work, and Dan and the other colleagues for the P53 work, and then also the team here. They contribute a lot to these studies and the funding support. I would like to thank you very much for listening to my presentation, and I wish you're safe and health in this pandemic period of time. Thank you.
Oh, thank you so much, Jinjun, for that very wonderful talk. So as a person who works in nano, I cannot emphasize enough on the importance of what Professor Shi has just said, because, like RNA and RNA-based medicine is actually, really, the problem is they are not stable. So if you have something that can prolong the stability from 5 minutes to 30 minutes to 1 hour to 5 hours, I mean, that's good enough to being translated into clinics. So thank you, Jinjun, for that wonderful talk. So next up, thank you so much. So next up, we have Professor Xiaoding Xu, and from Sun Yat-sen University. So let me introduce Professor Xiaoding. So he obtained his bachelor in chemistry and PhD in polymer chemistry and Physics at Wuhan University in 2006 and 2011. So after finishing 2 years of postdoc in China, he joined Professor Omid Faroukzad's lab in 2014 and worked at Harvard Medical School as a postdoctoral research fellow. And after 3 years, he was promoted into to become an instructor, also at Harvard Medical School. And then now he joined Sun Yat-sen Memorial Hospital and is currently a professor here. So Professor Xu is actually my direct senior. We worked together in Boston for 3 years with Professor Xi in Omid's lab before we joined the current position. So Professor Xu's research focuses is on the synthesis and use of polymeric nanomaterials for nucleic acid delivery and cancer therapy. So today, Professor Xu will share with us on the topic of nanoparticle-mediated long non-coding RNA silencing for effective cancer therapy. So please welcome him.
Thanks for Pierre's introduction. So good evening, everyone. I'm Xiao Ding, working at Sun Yat-sen Memorial Hospital. Today, I would like to present one of our recent work, nanoparticles mediate long non-coding RNA silence for effective cancer radiotherapy. So because Pierre has introduced me, so I will not introduce myself. So next I will go into my talk. So as we know, more than 90% of our human genes cannot encoding proteins, but can be transcribed into non-coding RNAs. These non-coding RNAs may execute their biological functions. Currently, it has been demonstrated that many non-coding RNAs can evolve intracellular signal networks and regulate the development and the progression of tumors. For example, the long non-coding RNA HTAL can promote the invasion and metastasis of breast cancer and lung cancer through the activation of several classic signal pathways, such as Wnt beta-catenin pathway and NF-kappa B signal pathway. Therefore, explore new functional non-coding RNAs and understanding their regulation mechanisms could provide new therapeutic targets for effective cancer therapy. So long non-coding RNA are class of long non-coding RNAs are class of non-coding RNA with a length more than 200 nucleotides. Link RNAs are widely involved in tumor genesis and progressions, and the regulation of their expression usually induce multiple biological functions. In general, long non-coding RNAs can interact with chromatin or interact with protein or RNA to regulate protein expression or regulate gene expression. Currently, many long-coding RNAs has been discovered, and they have been widely involved in tumor genesis and progression, such as promoting tumor cell proliferation, evading apoptosis, and improving angiogenesis. Due to their important functions, link RNAs has been now identified as attractive therapeutic targets for effective cancer treatment. And also now several clinical trials based on regulating link RNA functions are now ongoing. So radiotherapy is an effective treatment for many cancers, but radio resistance is frequently encountered in clinic. Ionized radiation can induce the generation of ROS, high-level ROS, and this ROS could cause DNA breaks or activate apoptic signal pathways to induce cell apoptosis. However, tumor cells usually adaptively generate a high concentration antioxidants and reduction agents such as GSH, which can scavenge the ROS and maintain the intracellular redox homeostasis for cell survival, and finally inducing radio resistance. Therefore, disrupting intracellular redox homeostasis through promoting ROS generation and or reducing RS scavenge has been long considered as an effective strategy to enhance radio sensitivity. However, radio resistance is a complex process associated with multiple biological changes that are regulated by intracellular signal pathways. Therefore, benefit treatment for radio-resistant cancer patients needed to include comprehensive consideration of multiple biological changes contributing radio resistance. So based on our formal consideration, we speculate that there are some link RNAs to involve radio resistance, and explore the key link RNAs would facilitate to reverse radio resistance. In addition, due to the generation of a high concentration of reduction agent, such as GSH under radiation, systemic delivery of siRNA using reduction responsive nanoparticles can synergistically reverse radio resistance through concurrently silence link RNA expression and scavenge intracellular GSH, leading to effective cancer radiotherapy. So along this principle, we in this work we collect the matched tumor tissues of 2 triple-negative breast cancer patients. Also, we call it TNBC patients before post-operative radiotherapy and after post-operative radiotherapy with local recurrence, and then employed link RNA microarray to analyze and compare their link RNA expression profile. In addition, we also construct radio-resistive triple-negative breast cancer cells and compare the link RNA expression file with the parental cell line. Finally, we found that 6 link RNAs are upregulated in both radio-resistant tumor samples and cells. We also use qPCR analysis, and the result shows that the link RNA AFAP1 and AS1 is highest in the radio-resistant triple-negative breast cancer cells. So to further validate the high expression of link AFAP1, AS1 is to further examine the high expression of this link RNA in radio-resistance TNBC. So we next this link RNA expression in 12 TNBC patients received post-operative radiotherapy. It can be seen in the figure. Compared to the patient without recurrence, higher link AFAP1 AS1 expression can be found in the tumor tissues of the patient with recurrence. In addition, through examining the link AFAP1 AS1 expression in 125 TNBC patients, we found that its high expression is associated with poor DFS and poor OS. In addition, in this 125 patients, there are 22 patients with local recurrence, and in these patients with local recurrence, high link AFAP1 AS1 expression is found. So we can from this data and conclude that the high AFAP1 AS1 expression is associated with the radio resistance of TNBC patients, and the expression level of this link RNA in tumor tissues could be used to predict the outcome of radiotherapy. So we next examine the intracellular location of this link RNA. We use the FISH, and the result shows that the most of the link RNA is located in the cytoplasm. Then we silence the link AFAP1 AS1 expression, use the lipofectomy, and the result shows that this link RNA silence can reverse radiosensitivity of radio-resistant cells, including low cell viability and more apoptosis. However, if overexpression of the link AFAP1 expression, the cancer cells show the radio resistance. So next, we want to explain why link AFAP1 overexpression induces radio resistance. So we use RNA pull down and then use mass spectral to identify the protein interacting with link AFAP1 AS1. As shown in this figure, beta-catenin is the specific link RNA binding protein. This specific interaction is further demonstrated by the RNA pull down followed by Western blot analysis. In general, beta-catenin expression is tightly controlled by the destruction complex in which the tumor suppressor protein axin act as a scaffold to interact with beta-catenin, APC, CK1, and GSK3 beta. In this destruction complex, CK1 and phosphorylate GSK3 beta can induce the phosphorylation of axin-bounded beta-catenin, leading to beta-catenin degradation. Therefore, we predict that link AFAP1-S1 might suppress the function of the beta-catenin destruction complex, and thus induce the release of beta-catenin to play its biological functions. To verify our prediction, we silence the link AFAP1-S1 expression and examine the expression of CK1, GSK3 beta, and the phosphorylation of GSK3 beta and the beta-catenin. We found that high link AFAP1-S S1 expression show low level of GSK3 beta phosphorylation and high expression of total and active beta-catenin. After silence this link RNA expression, there is no obvious in the level, no obvious change in the expression of CK1 and GSK3 beta. However, the phosphorylation of GSK3 beta is obviously improved. So this result indicates that the link AFAP1-AS1 can block the function of beta-catenin destruction complex by suppressing the phosphorylation of GSK3 beta expression, thereby leading to the release of beta-catenin and activating the classic Wnt beta-catenin signaling pathway to promote breast cancer cell proliferation and migration. So we further examine the dissociating constant between link AFAP1-AS1 and beta-catenin. We added different amount of this link RNA and then used the best plot to analyze the expression of beta-catenin. And from this figure, we can find that the KD between this link RNA and the beta-catenin is around 3.59 nanomole. So next, because link AFAP1 can activate the classic Wnt beta-catenin signaling pathway to induce the radio resistance. So we guess that, we think that systemic delivery siRNA to silence this link RNA expression could be a promising strategy to reverse radio resistance. So we previously developed a series of nanoparticles for systemic siRNA delivery, just like Professor Jin Junshi reported, including redox responsive, pH responsive, RS responsive nanoparticles. But considering the generation of a high concentration of a reduction agent, GSH, and radiation, so we chose reduction responsive nanoparticle for siRNA delivery and expect to synergistically reward the radio resistance through the concurrently silence the link AF-AP1 AS1 expression and scavenge the intracellular GSH. So this reduction responsive nanoparticle platform is composed of reduction responsive polymer. We call it PDSA, and outer PG shell, which could prolong the blood circulation of the nanoparticles. As shown in this figure, after loading the link AF-AP1 AS1 sRNA, the nanoparticle shows uniform morphology with a size around 110 nanometer. Use this nanoparticle to treat the radio-resistant breast cancer cells. More than 70% of the link RNA expression could be suppressed at sRNA dose around 30 nanomole. With this efficient link RNA silence, beta-catenin expression is obviously suppressed. More importantly, because these nanoparticles are reduction responsive, they can scavenge the intracellular GSH, and thus the GSH expression always maintains a low level under radiation. With this link RNA silencing and the GSH scavenge, the nanoparticles could synergistically reward the radio resistance of breast cancer cells. As shown in this figure, the nanoparticles are the most effective in inhibiting the survival of the breast cancer cells on the radiation compared to the single GSH scavenge or single link RNA silencing. So with the above promising in vitro experimental data, so we finally evaluate whether the reduction responsive nanoparticle can deliver link RNA, sRNA, and enhance the therapeutic outcome of radiotherapy. So to this end, the nanoparticle was intravenously injected to the breast cancer cell xenograft tumor-bearing mice once every 2 days. After 3 consecutive injections, the tumors show favorable sensitivity to the radiation, and the tumor growth is obviously inhibited. As we know, radio resistance usually leading to enhanced local invasion and metastasis of cancer patients. Therefore, in this work, we established a metastatic model using the luciferase expression radio resistant breast cancer cells to further demonstrate the ability of our reduction responsive nanoparticles for sRNA delivery to enhance the radiotherapy effect as we expected, with the concurrent link AF-AP1 silencing and GSH scavenge, the nanoparticle treatment followed by radiation shows the strongest ability to reduce the metastatic tumor burden. So a brief summary. So in this work, we have identified a functional link RNA. We call it link AF-AP1 AS1 in radio-resistant TNBC patients, and this link RNA could induce radio resistance via activating beta-catenin signaling pathway. Silence this link RNA expression using reduction responsive nanoparticles could synergistically reward radio resistance. So I would like to thank the support from several grants such as the NSFC and the 1000 Talent and the Department of Science and Technology of the Guangdong Province and the Guangzhou Science and Technology Bureau. And also this work, we collaborate with Professor He Yao and Professor Hu Hai and also Professor Pierre. And also I would like to thank the students, Dr. Zhou Feibi and Dr. Qingjian Li. Finally, thank you for your time to listen to my talk. Thank you again.
Yeah, thank you so much, Xiaoding. I think it's hard for Xiaoding actually to talk in English right now because we have been back for maybe 3 years now. I think it's yeah. So Xiaoding has not been presenting in English. So we really thank you for today. And so this is a classic example of really an integration, a really good integration, because before we came back, I think Xiaoding was working on polymeric nanoparticles, but not in a significant way of integration with the clinics. But when we come here, when we arrive at Sun Yat-sen Memorial Hospital, and we found out that actually we are the pioneer of long non-coding RNA, which Professor Xiaoding has mentioned. So knowing that and the basic research that they have done, and then Professor Xu just took it to another level. So he took the link RNA and then encapsulate in the nanoparticle, and then now we try to deliver this to solve a clinical problem that is radio resistance. So I think this is a very classic example of why we should integrate different majors in our work. So thank you, Professor Xiaoding. So next we have Professor Zhen Yuan. And so before that, let me introduce Professor Yuan. So Professor Zhen Yuan is an associate professor with the Faculty of Health Sciences at University of Macau, which is very close to Guangzhou. And he's also the director of Center of Cognitive and Brain Sciences. So before joining University of Macau, he had worked as an assistant professor with the Arizona State University and research assistant professor in biomedical engineering department with the University of Florida. And his academic investigation is focused on cutting edge research and development in laser, ultrasound, and EEG fMRI related biomedical techniques, as well as their clinical and preclinical applications in neuroimaging and neurosciences, and particularly in molecular imaging and cancer. He has received national and international recognition through more than 200 publications in his field. So today, he will be sharing with us on the topic of photoacoustic imaging guided cancer diagnostic. So let us welcome Professor Yuan.
Okay. Thanks, Pierre, for the kind introduction. Dear colleagues, friends, and audience, good evening. Thanks for being with us. I think I need to share my screen. I think, okay. Can you see my PPT? Okay. I think our speaker actually have delivered a wonderful talk. I think across biology and chemistry, nanomedicine, and also technique, right? It's wonderful. I think I learned a lot from from them. Their work really impressive. And okay, today I will be talking about the photoacoustic imaging guided catheter diagnostics. I'm Zhen Yuan from the University of Macau. And actually, my research actually focused on three perspectives. The first is focused on the first one is technique, right? Biomedical optics is imaging techniques. We develop a lot of imaging setup for brain imaging and also cancer imaging. The second one is the optical molecular imaging. We also develop a lot of probe, including nanoprobe for preclinical study and also very basic biology study. The third one is actually I started my this kind of direction after I joined UM. Actually, I have been working on that for nearly 7 years. It's a neuroimaging. I think I use the EEG, FNIS, and fMRI to do a lot of cognitive neurosciences and also AI, artificial intelligence. So firstly, I give a brief introduction to my lab. Actually, my lab focuses on two perspectives, right? One is molecular imaging, particularly optical molecular imaging. The other is neural imaging. For optical molecular imaging platform, we have a bunch of homemade and also commercial imaging setup available in my lab for in vivo imaging across different scales. For example, in the submicro scale, we have the fluorescence microscopy and also optical resolution photoquantum microscopy. And over factory also have the super resolution and also the two photon microscopy imaging setup. In the macro level, we have the OCT and also acoustic resolution photoquantum microscopy imaging setup. And at the tissue level, we have the homemade photoquantum imaging setup and commercial ultrasound imaging setup, and also macro CT with, and also the fluorescence molecular imaging setup for small animal imaging, particularly the mouse and the rats. Besides the neural imaging platform, we also have molecular imaging platform. We also have the neural imaging platform. Our neural imaging platform consists of 4 cores. The first core is the neural imaging core. It's considered a lot of 12 FNIRs, functional near-infrared spectroscopy optical neural imaging setup. And we also have 12 EEG setup, including the FMI compatible EEG setup. And the second core is the brain intervention core. We have the TMS, RTMS, and ultrasound interventional setup. And the third one is the core is we call the behavioral data collection core. It's actually this kind of setup useful to collect the behavioral data from animal to human being. The last one is the high performance computing core. We have the high performance computing center in the it's a part of China high performance computing. We call the cloud. Its southern part is located in Macau, actually, a southern part of China. And recently we entered the 3T FMI, Siemens Prisma 3T FMI, basically used for the animal and also human being neural disorder and other cognitive neuroscience studies. Here actually focus on the language study. It's really social science, but I'm very interested in this kind of study. And also you involve the Portuguese, the Chinese, and English translation, and also the brain cognition across the bilinguals, and also the addiction, behavioral addiction, such as gambling, gaming, and also decision making. And we also child development, aging, and brain disorder, and also, of course, also technically related to the neural imaging, particularly also the optical neural imaging, right? Is my strength actually. Then I give some introduction to the background of this presentation. Actually, this presentation is for cancer therapeutics, right? We know that cancer actually is one of the leading cause of the mortality and also mobility around the world, right? So if we use the imaging technique to achieve cancer early detection, also achieve the imaging-guided therapy, we can improve the survival rate of the patient, and also can actually reduce the side effect during the treatment. NIH published data show that if breast cancer, right, is detected under the stage 1 or stage 2, right, the 5-year survival rate can be up to 100%. So that's why we need to do the cancer early detection and also achieve the imaging-guided cancer treatment. And we know that for traditional biomedical imaging techniques, such as the CT, MI, right? It's kind of a structural imaging technique cannot achieve cancer early detection because of the low sensitivity, right? No functional information. And also very hard for this kind of imaging technique to achieve simultaneous cancer treatment and also cancer detection, right? Additionally, actually for cancer treatment, we have the surgery, radiotherapy, and also chemotherapy. Recently, very interestingly, we know that phototherapy, right, is attract a lot of attention, incentive attention, because it's non-invasiveness, right? It's a technique. It also can be carried out locally, right? Based on the locally selected area, cancer, very target area, and also it's all lower have the lower drug resistance, right? And also lower side effects. That's the advantage. Among other image techniques developed that have been to resolve the basic limitation of the lower sensitivity of CT and MRI, right? Photoquantum imaging is particularly very interesting and also promise, right? It's a very promising technique because it can achieve simultaneous functional, molecular, right? And also structural imaging. It can also achieve the neural imaging, functional neural imaging. That's a very interesting technique, and also with high resolution, high contrast. So I just show how the photoquantum works. When we find the lead pulse, right, the photon will be propagated together in the beginning, then the photon will be diverged in the diffusion domain. When the photon reach 2 or 3 up to 1 millimeter, right, to reach to the diffusion domain 2 or 3 centimeter, right? So the photon will be absorbed. Some photon will be scattered, right? Some photon will be transmitted from the surface of tissue. When the photon are absorbed by the tissue, it will generate heat. So the heat will generate acoustic wave. So the wave will propagate to the together. So acoustic wave will propagate to the surface of tissue, where they are detected by the ultrasound array, right? So based on the measured photoquantum signal, we can actually achieve the photoquantum functional imaging, right, to recover the functional information with high optical contrast. So photoquantum show the advantage where the challenge, particularly for the label-free photoquantum imaging, right? For label-free photoquantum imaging, generally we use the visible light or the first window light to illuminate the animal or the tissue, right? So generally it will reduce the penetration depths. So that's the one issue. The second issue that photoquantum imaging, label-free photoquantum imaging, they use the optical contrast. Optical contrast is very good for imaging the soft tissue, including the blood vessel, right? But it's very hard for the photoquantum imaging to recover the functional and the structural information of the hard tissue, for example, bones, right? And other soft tissue with lower contrast, right? So it's very hard. So we have to use the multiple contrast and there multiple imaging setup to resolve this kind of issue to recover most of the tissue, right? From soft tissue, hard tissue, right? And also including the blood vessel. And also label-free photoquantum imaging technique is sufficient for high sensitivity, cancer detection, because so that's why we need to use a nanoprobe, a contrast agent, to improve the resolution and contrast and the sensitivity, right? And also achieve, molecular imaging gadget kind of treatment, right? And so that's a bigger challenge that is here, right? You can see that we need to use the multimodality imaging technique, right? We need to develop the multifunctional nanoprobe as a contrast agent for multimodality molecular imaging, and also this kind of nanoprobe also serve the phototherapy agents for photodynamic thermotherapy agents, right? Sometimes it can also serve the chemotherapy agents. Depend your design, right? Depend your requirement of your the treatment, and we also need to actually perform the second near-infrared window photoquantum imaging. That means because the second window can near-infrared light can improve the penetration depths of the photoquantum imaging technique. Right now, actually, basically, there are 3 categories of the photoquantum contrast agent for cancer theranostics. The first is small molecular, right? It's Overlap has also done a lot of work on that. The second one is inorganic nanoprobe. The third one is organic nanoprobe. Today, I will show 2 very representative nanoprobe for multifunctional near-infrared sector near-infrared nanoprobe for the catheterinotics. The first one is the organic, right? The second one is organic nanoprobe. The organic probe we use the polymer dots. Because they have high good, biocompatible property, and also it's also has a small size, and also has a lower side effect, and also has high optical absorption property. Can be used for simultaneous photocoded image, and also can serve the current agent for simultaneous photocoded imaging, and also photothermal and photothermal agents. The first one is we call it is organic nanoprobe. We call it is GA functionalized carbon sulfur nanoprobe for dual mode photocoded image and MRI imaging near the second near-infrared window. So this image show is a schematic show of the how the GA functional BSA modified carbon sulfur nanoprobe can be used for the dual model molecular imaging, and also for the cancer treatment. So this basically BSA with a cation anion, right? With under the condition of the pH value higher than 7 and also heat, right? We will produce the triangle BSA modified carbon sulfur nanoparticles. Then GA will integrate to the lead nanoparticle to generate the GA functionalized carbon sulfur nanoparticle, right? This nanoparticle has multiple function. It can be used for, so the GA actually serves the current agent for MR imaging, right? The carbon sulfur nanoparticle, they have high optical absorption, with is very broad absorption band, right? Can be used for the optical or photocoded imaging in the visible window, and the first window and the second near-infrared window. So it has a lot of functions. So then we do the we show the how we do the calculation of the conjugate functional carbon sulfur nanoparticle, right? So we can see that this is the TM image, right? Show the size and also the shape of the nanoparticle. This one actually show the absorption spectra, right? For the produced nanoparticle. We can see that the peak is in the second near-infrared window. And this one actually show the optical spectra before and after they got conjugated to the carbon sulfur nanoparticle. And we also do the dynamic lattice scattering measurement, and also this and we also do the in vitro test, right? Show that actually the probe, right, really can improve the contrast agent of the photocoded image and MRI image. You can see that the photocoded signal and the image, right, increased with increased concentration of the nanoprobes. And also the nanoprobe actually show that it has a lower toxicity to the cells. The cell viability is the test show the efficacy of these nanoprobes. Then we do the image, right? So this is the photocoded image, and this is the MRI imaging. So photocoded image, we only image the tumor area, right? We can see that after we inject the nanoprobe through the TIN, right? Intravenous TIN injection of the conjugated carbon sulfur nanoparticle, right? We can see that the photocoded signal image, right? The current agent actually is enhanced from before the injection and after post-injection, and 6 hours, 12 hours, and also 24 hours, right? So basically the photocoded signal also show the enhancement increase, the signal increase, right? And the different time points. Simultaneously, we can also see that photocoded signal also show the increasement, right? Increase. We can see the the signal also increased from before the injection, after the injection, right? And the counter agent also, we can see that is significantly improved after we inject the counter agent. Actually, we can also see that for MR imaging, right? If you just do the if you do the label-free imaging, right, it's easily to miss the tumor, right? Because generally the contrast agent for the tumor, the contrast for the tumor area, right, is smaller than the surrounding tissue. So that's why I need to do the multimodality molecular imaging, right, to really improve the sensitivity, the detection sensitivity, right, to improve the sensitivity, resolution, and also basically sensitivity for cancer detection and also to guide the treatment. Histology analysis also be performed, right? We can see that it basically we can see the nanoparticle basically show no damage to the normal tissue, right? We compare to the control group, right, PBS. So that show the efficacy and also show the performance of the developed multifunctional nanoprobe. Again, we also show a very representative nanoprobe, organic nanoprobe, right? That's the PDOTS. Recently, we published this work. This is a new work we just published actually last month. We know that actually organic nanoprobe, they show a lot of advantage, right? It's a high absorption, very good, excellent biocompatibility, right? To tissue, right? So it's a lower toxicity, right? And high absorption surface. So it has a lot of advantage compared to the inorganic nanoprobe. So we can see that this is a structure of the DPP, BTD, TD organic probe, right? So that show the second image show how the DPP, BTD, TD nanoprobe are produced and used for the catheteranotics. For this project, actually, we produced two PDOTs. The first one is actually, we call the S-PDOTs. It has ultra small size, only 4 nanometer. It's easily can be extracted quickly, cleaned from the body, right? And no, actually, it will not store in the body. No damage to the, to the normal organs. The second one is we call the L-DOTs. This kind of PDOTs have the diameter of 25 nanometers, right? So we want to do the comparison to show really is the ultra small nanoparticle can improve the metabolic capability, and we want to inspect whether you can quickly clean from the body, right? And so this is show the dynamic light scattering signal image figures, right? We can see that the size, right, around 4 nanometer. The second one, PDOTs, we call the L-DOTs, is sized around 25 nanometer. And the theta potential, we also did the theta potential, and also the optical spectrum show that the peak of the optical absorption is located in the second near-field window, right? So we can see that the peak. And we also tested the photothermal property of the derived ultra small nanoparticle in the second near-field window, right? So this one show actually the photothermal heating curve, right, with the different power of the laser. And this one show that the photothermal image, right? Photothermal actually image and photothermal image and with different concentration of the nanoprobe. And then this show the temperature evolution of the ultra small nanoparticle with different concentration, right? And also the circles, right, on and off for the photothermal therapy. And then we also have this kind of photothermal performance, right, to compare with water, right? Water can show the photothermal property of the PDOTs. Then we do the in vitro photothermal therapy test, right? Photothermal test of the cancer cell based on the derived ultra small PDOTs. So basically, without laser, right? Without laser irradiation, we basically did, we can see that most of the hairless cancer cell, right, when they incubate with the PDOS, basically it is show no damage or toxicity to the cancer cell, right, during 24 hours and 48 hours. But when we use the laser irradiation, right, and also without irradiation at the control group, we can see that most of the cancer cells were killed. And also confocal imaging also performed with nanoparticle, with laser or without laser, right? So without laser irradiation, when the hair cell were incubated with the nanoparticle, right? So basically most of the cancer cell will survive, right? So the green one actually show the live cell, the red one show the dead cell, cancer cell, right? But when we use the laser to remove the cancer cell without any nanoparticle, right, it's basically also the cancer cell survive. But when we use the with the laser irradiation, and then we increase the nanoparticle concentration, we can see that most of the cancer cell began to die. Some become dead, right? Some particularly with this concentration, right? Most of the hairless cancer cell were dead after irradiation, right? Within 5 minutes. Then in vivo test, photocopic imaging tests were also carried out. We can see that basically photocopic signal increase with the increased concentration of the nanoprobe, right? And we also compare the photocopic imaging capability between the two PDOS. One is ultra small PDOS, we call it S PDOS, right? We can see that this is the photo imaging, the tumor tissue, right? Before the injection, and then after post-injection, and 1 hour, 0 hour, right? 1 hour, 2 hour, till 24 hours. And then we also do the photocopic imaging for the big size LDOS, right? 25 nanometer LPDOS, right? We can also see the photocopic signal increased, right? But we can see, but it's different, right? For the ultra small PDOS, the signal can increase quickly, right? We can after 2 hours it reach the peak, right? But for the large size nanoparticle, right, it can basically take a couple of hours to reach the peak. And we also do the photothermal therapy, right? It shows the photothermal ion image, right? Thermal image for after we inject the SPDOS, right? And do the photothermal treatment. And the second group will show actually the contrast group, right? Didn't show the temperature increase. This one show the temperature change, the image, right? And then also compare the temperature evolution between the different groups, right? Control group PBS, right? PBS with laser irradiation, SPDOT without laser irradiation, and with PDOS, SPDOT with laser irradiation, right? We can see that only this group, ultra small SPDOS with laser irradiation, it show the temperature increase, right? To up to 55 centigrade, right? And we also do the photothermometry for different groups, right? The 4 groups. And we take the photography and day 0, day 6, and day 14, right? And then we also compare the tumor volume based on different treatment, right? We can see that only the ultra small PDOS group with near-infrared, second near-infrared laser irradiation show they reduce the tumor volume, right? The red one, right? And also the body actually weight didn't show significant change across the different groups. And finally, actually, I want to show that actually the PDOS actually really no damage, right, to the normal tissue, normal organs. And it can show, this is the large size PDOS, this is ultra small PDOS. We can see that after 3 days, right, the ultra small PDOS can quickly extract from also the organ or the body, right? So, and also validated by the blood signal. We use a lot of blood Lutein index to show the metabolic property of the P dot, ultra small P dots. So I want to quickly finish my presentation because it's very late. I think okay. I want to take any question. Oh no, we don't have question, right? Okay. Welcome to visit our campus, right? It's Macau is a very nice place, very multicultural European style city in Asia. I think is and people are very nice, and we don't have the so far we don't have the virus, right? No people got infected so far. Previously, 4 months ago, we have several patients, but actually it's imported from other area, but all of them are recovered right now. So it's a very safe place. Okay, thank you.
All right, thank you so much, Professor Yuan, and thank you for your presentation. Also, thank you for your invitation. So everybody, please go to Macau and find Professor Yuan, okay? He will be very happy to be our host. So we have come to the end of today's presentation, and I would like to thank all speakers for today. And for myself, I've learned a lot, and the take-home message today, I think, is that there are many ways to solve a problem. And the solution might be a simple molecule that can be found around us. And although nanomedicine is not a magic bullet that could solve all the problems, but by integration of various fields, I think it will become one of the most effective ways that could interfere with many points or pathways in various diseases. So we applaud all researchers and scientists working hard even to this day to find new interventions for the betterment of human mankind. So we thank you for that. Now we have come to the conclusion of today's conference. We thank you to all the speakers for their time in sharing with us their groundbreaking works. We also thank all of you who are joining us today online. We hope to see you again in our future conference. And with that, we wish you a good evening and see you again.
Okay, thank you. Bye-bye.